Today we're going to be looking at the activity Rare Genetic Diseases. This activity has two main learning objectives. So first, students will apply their knowledge of DNA and heredity to diagnose real-world rare genetic disease case studies. And second, students will work in collaborative teams to collect and analyze the data required to communicate scientific information during a patient-doctor interaction. These are the materials that you will need to do this activity. You'll need the team sheet, the bioinformatician sheet, the geneticist sheet, the family doctor sheet, and the gene cards, as well as a post-it book, dry erase markers, and a pedigree tool. Let's work on Rachel's case study together. First, we'll assign positions. Write your name on a sticky note and place it on your roll. Now let's explore what each of the roles do. First up is the doctor. They communicate with the patient and the geneticist. They identify the body systems that could be impacted by the symptoms. They describe the family history by looking at the pedigree chart and use the drawing tool to help. Next up, the Gene Master. The Gene Master uses cards to assist geneticists and bioinformaticians. The Gene Master also helps the bioinformatician to understand which alleles are bad and which alleles are good. They also help the geneticist to connect patient symptoms to their genetic bases. Next up, the bioinformatician. The bioinformatician communicates with the Gene Master and the geneticist. They complete the table to determine which alleles are likely to express as disease phenotypes in patients. And lastly, the geneticist. They communicate with the Gene Master, bioinformatician, and the doctor. They match the symptoms presented by the patient with underlying genetic causes and recommend a course of action to the doctor. Now, let's explore a case study together. This is Rachel. Imagine you are a doctor and she showed up in your office. What questions would you ask? What do you see? What might be the cause? Rachel shows you a picture of her family. Some of her siblings and parents share her condition. This suggests that it's a genetic component. So first, let's look at Rachel's case study as a doctor. Let's take a look at Rachel's symptoms. You'll notice that she has blue skin, and she describes that she has difficulty in school, which means that her neurological system and another system is impacted. Now, let's look at her family history. We'll notice that she has an affected father, as his pedigree is completely filled. Her mother is a carrier, as her pedigree is half filled. You'll also notice that she has an affected sister. So now, as a doctor, you'll recommend a whole genome sequencing. So as a bioinformatician, you'll have a report that indicates which genes may be the cause of these problems. So now you'll talk to the geneticist to cross-reference which genes you think are the problem. In this case, 2 genes matched, and we removed the other one. So now the geneticist will ask the Gene Master to provide more information on what the genes do. First, the geneticist will want to know if the symptoms match. The symptoms of the CYB5R3 gene match the symptoms of the family doctor's report, while the DLL4 symptoms do not match. The geneticist will record this in the symptoms column. Now, with the help of the Gene Master's cards, the bioinformatician will fill out the reference allele, which is the non-disease causing allele, the alternative allele, which is the disease causing allele, functional, which is the type of mutation, and the inheritance pattern, which is the way that it is expressed for each possible gene. Now the bioinformatician will collaborate with the doctor to compare the phenotypes, which is what we can see on the pedigree, to the genotypes, which is what we see in the report. So in the first case, the father and mother have the same genotype, but their phenotypes are different, which means that this is not an inheritance match. In the second case, the child and father have the same genotype and the same phenotype, whereas the mother has a different genotype, and on the pedigree we see that she also has a different phenotype. So yes, this is an inheritance match. So now the bioinformatician will talk with the geneticist to determine which genes had an inheritance match. So here we see that the CYB5R3 gene did have an inheritance match, while the DLL4 gene did not. So now, the geneticist will go across each row to see if both the symptoms match and the inheritance match, and they will mark this as the candidate gene. In this case, the CYB5R3 gene is a candidate gene, whereas the AKAP1 and the DLL4 genes are not. So now the geneticist, with the help of the Gene Master's card, will determine that the CYB5R3 gene causes methemoglobinemia. So now that the geneticist has determined the candidate gene, the doctor can make a diagnosis. Following the diagnosis, the doctor will outline the next steps. So in this case, the family doctor will tell the patient that they have a diagnosis of methemoglobinemia, and the next steps are oxygen therapy. So now that Rachel has her diagnosis, we can switch up the roles and use a similar approach to tackle another patient's case study. If you want to learn more about the patients that this activity was modeled after, check out the following links. Now that we've outlined this activity, check out other Genome BC resources and activities here.